Hello, everyone, and welcome to today's webinar, Development of an Automated Online Flow Cytometry Method to Quantify Cell Density and Fingerprint Bacterial Communities. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I would also like to point out that there is a survey question in the interface on the bottom left of your screen. Please make sure to submit your response during this broadcast. I would now like to introduce our speakers, Dr. Konstanze Schiessl, Chief Scientific Officer, onCyt Microbiology, and Juan López Gálvez, Ph.D. researcher. For complete biographies on our speakers, please visit the Biography tab at the top right of your screen. We will now begin our presentation.
Hello, I'm Matthew Goff, Senior Product Manager for Cytoflex Flow Cytometers at Beckman Coulter Life Sciences. Today, it is our privilege to promote cutting-edge research that appears at the intersection of automation and flow cytometry. At Beckman Coulter Life Sciences, we are constantly seeking new ways to address scientific inquiry by focusing on accessible, robust technologies which give researchers the tools required to create rigorous and reproducible data. The Cytoflex Flow Cytometer featured today is a scalable, affordable solution for your lab. Whether you need as few as 4 channels of detection or as many as 23, we have a solution that will fit your needs while allowing you to grow. Users of the Cytoflex analyzers have told us that they enjoy the sensitivity of the hardware, as well as the ease of use and accessibility of the software interface. While operation is simple, this powerful tool can be adjoined to advanced automation solutions, such as those that will be presented here. I'm happy to introduce today's presenters, Chief Scientific Officer at onCyt Microbiology, Konstanze Schiessl, as well as Ph.D. researcher, Juan López Gálvez, to teach you more about the development of an automated online flow cytometry method to quantify cell density fingerprint bacterial communities.
Hello, everyone. Thanks for taking the time to listen to our webinar. Thanks for the kind introduction and the invitation. So Juan and I will be guiding you through the webinar today. We will be diving into the world of microbiology, and we will tell you about the great power of flow cytometry for achieving 3 things that are hidden in this kind of complicated title here. So the 3 things are counting bacteria, differentiating bacteria in a mixed community, and tracking their dynamics over time. These 3 things, they may sound simple, but they're actually quite difficult to accomplish. And we spent some time to develop a method for achieving this, and we will tell you about this today. So why did we dedicate our time to this? What is it useful for? Microbial communities, they're really fascinating. And by working together, bacteria can make amazing transformation processes that physical or chemical methods might not always achieve. So bacteria can, for example, transform waste into valuable products like biofuel or bioplastics. And in order to make these processes more efficient and to optimize them, monitoring technology is needed. And this is exactly what we did here. So we established a monitoring technology that will help industry and researchers to make such processes more efficient. Okay, sorry about this. There's a little glimpse in the slides. We'd like to introduce the team that was working on this. So on the top, you see that we are all part of the Promicon consortium. So this is a Horizon 2020 project of the European Union, and it's all centered around microbial communities and the transformation processes they can achieve. Here for this project, there's a team of 2. That's the company onCyt that I'm working for. We are located in beautiful Zurich in Switzerland, and the people who worked on this project specifically was me, and then our CEO and founder, Michael Besma, and Marisa Silva, who you see in this picture here. We are a startup company. We come from the Aquatic Federal Research Institute here in Switzerland, and we develop automation technology that can be used with the Cytoflex. So maybe Juan, you can present your group.
Yes. First, I would like to thank Matthew for the nice introduction. And yes, I am working in the Helmholtz Center for Environmental Research. This is based in Leipzig, Germany, and I work specifically in the working group for flow cytometry. The contributors from our group that helped us to develop this publication was myself, our group leader, work group leader Susanne Müller, another Ph.D. researcher called Carmen Bruckmann, and our department leader Hauke Harms.
Good. I'll give you a quick outline of what's to come. So in the first part, Juan will explain a little bit about the power of flow cytometry for microbial quantification. Then I will take over to tell you about the power of online flow cytometry for understanding microbial dynamics. And at the end, we will focus on this paper and explain more details here. So Juan, over to you.
Yeah, like Konstanze said, first we will talk about the power of flow cytometry for microbial quantification. Flow cytometry is already a technique that is widely used for the analysis of bacterial communities, both artificial and synthetic. And there are several papers and study cases in which this technique was used to determine ecological variables of bacterial communities. And I would like to give you some examples about this. For example, here we have this publication published in 2021. This publication analyzed the community to resolve the predatory interactions between bacteria in a high diversity system. Second example was this publication published in 2022, and this deals with the stabilization of microbial communities by loop mass transfer. In this case, flow cytometry was used to determine these bacterial communities in the looped mass bioreactor. Another good example is this one published in 2020, and this deal with the study of intestinal microbiota to determine the community structure inside an organism. I just gave you three examples, but there are many others where flow cytometry was used to determine the bacterial composition of several different communities. However, all these examples that I showed were done by flow cytometry, but in a manual manner. This means that the sample taken and the preparation is performed by an operator. This, of course, brings several limitations. For example, you have a limited sampling. Second is that you have relatively long treatment times for both fixation and staining. When a procedure is done by an operator, this is prone to human error. And for these reasons, there's a big window of opportunity where an online automated procedure can be used for the sampling, pretreatment, and measurement of samples consisting of bacterial communities using flow cytometry.
Okay, so this is a nice introduction to the second part. If you have a system of interest, it can be anything. It can be a bioreactor, for example. The bacteria in there, they are not stable. They will fluctuate over time, which is just the nature of biology or of any process. And these fluctuations can be really important in order to control the process, to see if something is going out of hand, so to monitor it. And there are not many methods you can use to actually track these processes. So often researchers just take a manual sample, like Juan said, I don't know, maybe once every day. And if you take it at the wrong times, as you see here in the red line, maybe you think there's no dynamics going on at all, while in the background, the real dynamics are hidden just because of the inadequate sampling and analysis method you use. So many microbial quantification methods, they do not allow you to do high frequency in 24/7 analysis, and important process dynamics are simply missed. And flow cytometry can actually help with this if you run it in an online way, as we will explain later. So data you can see here, you can collect this type of data with the online flow cytometry method. Flow cytometry, it's really important in this context because many other technologies, they are just limited. They will not be able to give you this high frequency insight. I mean, cultivation is one extreme. If you grow bacteria on plates, it can take days until you get one data point. So there's no way you can get an insight into a microbial process very fast. And like Juan said, it's, this method, it's very error prone. Microscopy, it's the same story. Takes forever, low throughput, low statistics, very labor intense. Also not a way to get really at good microbial quantification, let alone at microbial dynamics. Molecular techniques also, they take a lot of time, require a lot of know-how, and not all of these methods really give you the quantitation that you might want to have. So flow cytometry, it's very fast. It's universal because all bacteria can be measured. They don't need to be grown. And this really makes it a very suitable method for quantifying bacteria in a rapid way. And we've seen over and over that bacteria fluctuate everywhere you look. There's never a stable line. So I just want to show two examples. It's not even so much about the axes. You will see on the y-axis we have concentration, certain types of concentrations, but it doesn't really matter. What you should focus on is the fluctuations you see here. So we've measured in all kinds of systems, in tap water, bioreactors, cooling water, water reuse facilities, and we never see a flat line. Microbes always change, and they often change with the process. So here in tap water, you, for example, see a sudden increase in the concentration. And this is because there was a stagnation period before. It was the weekend, people didn't really turn on the taps. Then you turn on the tap and then there's a flush of bacteria out of the system. And this type of data can really give you insight into controlling a process. This is the second example I want to show. It's really interesting because you see these sharp drops in concentration. This is from a cooling water system, and the drops come because biocide is added. And of course, the bacteria die very rapidly, but you also see they grow up very rapidly again. So you see it's a system that changes very fast. And if you imagine taking a sample here once a day, you will completely miss the dynamics of your process, and you will miss opportunities to improve your process. Okay, and now back to Juan for describing a bit more detail of our publication.
Yes, in this publication that was published in this year, 2023, we developed a new automated online flow cytometry procedure to quantify cell density and also to fingerprint bacterial communities. The objective of this work was to develop this procedure that couples the Cytoflex, the OC300 automation device that was developed and produced by onCyt, and the FlowMe version 2 bioinformatic tool to automatically obtain and analyze flow cytometry data of bacterial communities. Here is a visual representation of the objective. We have the onCyt device, which samples and pre-treats the bacterial community. Then, after the sample is treated, is sent to the flow cytometer to be measured, and after this, the data is automatically evaluated by the FlowMe bioinformatic tool. And this all together comes And this all comes together to have an automated cell fingerprinting for monitoring of consortia. So why was this important, or why we set this objective? Yeah, the accurate control of a bacterial community is necessary to drive the community into producing a product of interest. When we use an automated flow cytometric procedure, we can get fast and accurate information about the current state of the procedure without long waiting times or preparation times. And in this manner, flow cytometry acts as a real-time sensor for the control of any bacterial community that grows in a bioreactor. So how does this new procedure work? First, we have our bacterial community. This is automatically sampled by the OC300. After it's sampled, inside the device, the bacterial community is automatically fixed and stained. After the fixation and staining is performed, the sample is sent to the Cytoflex to be measured. And after the measurement is completed, there's an automatic data evaluation, and this all gives us a cell count and fingerprinting of this bacterial community. So here we have an overview of the automated procedure and how it works inside of the device. First, the sample is diluted in PBS to bring the cell optical density down to a treatable amount. This is moved to chamber 1 inside of the device. Then the sample is moved also to chamber 1 to be diluted. After this, all the reagents necessary for the fixations are taken in and sent to chamber 2 in order for the sample to be fixed. The reagents used for the fixations were sodium azide, sodium chloride, and ethanol. After 10 minutes of fixation, sample is moved to chamber 3 along with the DAP is stained in order to be stained. The staining takes 10 minutes. And after all of this is done, there is a final dilution in ultra-pure water, and the sample is sent to the flow cytometer to be measured. So first we needed to do was to validate the automated procedure, and the first step was to assure the self-cleaning capabilities of the device between each measurement. Self-cleaning is of course very important for the accurate monitoring of bacterial communities, as we want a sample that is clean from the previous sample, so the previous sample does not affect the cell concentration of the next sample that is measured. We tested a cell community conformed of 3 different bacteria. You can see them here. We have Penobacillus polymixa, Cochlear rhizophila, and Stenotophomonas rhizophila, and we tested 4 different cleaning solutions. We tested firstly as a cleaning solution only pure water. Then we tested also the flow clean agent reagent and several dilutions of them, a 10% dilution of them and a 1% dilution of them. And what you have to take from this table is that the best performing cleaning solution was the 1% flow clean agent. This evidenced by the low carryover number that we have from sample to sample. So because of this reason, we used always this cleaning solution in every future experiment. Next step, after we validated the self-cleaning capabilities of the device between measurements, was to validate the stability of measurements. For this, we used a bacterial community consisting of the same bacterial strains, and these bacteria were prefixed in a polyformaldehyde and ethanol fixation procedure to assure that there are no biological changes while the sample is being measured over a period of 35 hours, as you can see. We can see on the y-axis here, we can see the cell concentration of cells measured per each strain. And in this y-axis, we can see the forward scatter of the C strains as as time passes. We can see that the cell concentration remains constant for most of the time, as also the forward scatter, except at the final points of the measurement with when measuring the Penobacillus polymixa strain. So we were wondering why this change in cell concentration, why this increase in cell concentration and decrease in forward scatter. So our main hypothesis was that because polymixa tends to form clusters of cells, these clusters of cells are separated by physical forces as the sample is stirred and time passes by. To corroborate this hypothesis, we use the FlowEmi bioinformatic tool to analyze the different subcommunities consisting or that make up the Penobacillus polymixa, and we found out that there is an increase in the cell concentration. An increase in the cell concentration of the communities consisting of one chromosome number and two chromosome number, here marked as C1 and C2, as well as a decrease in the concentration of the subpopulation consisting of clusters of bacteria with 3 or more chromosome numbers, here marked as CX. With these results, we evidenced that these changes that we see in the cell concentration of polymixa are not caused by any biological change of the bacteria, as it is expected, because this bacteria is already prefixed, and is not also caused by any changes introduced by the procedure itself, but just rather by these physical forces separating the clusters of cells into cells with lower cell chromosome number. With this, I come to the conclusion, and this new procedure was successful in providing valuable information about the current state of any bacterial community. This is all done automatically without the need of an operator. This gives us more frequent samples with sampling. With this procedure, we can have a new sample measured every 45 minutes. This with the inclusion of the automatic evaluation tool, we have a faster analysis of data, and of course, because it's all done without an operator, we have an avoidance of human error. And the idea of the future plan is to use this new procedure to monitor and control a productive microbial consortia that's growing in a bioreactor and increase the yields based on the information obtained by this automatic flow cytometry procedure.
Okay, thanks Juan for telling us about the procedure. Now we're very happy to take any questions from the audience, and thank you all for listening.
Thank you, Matt, Konstanze, and Juan, for your informative presentations. Now we will start the live Q&A portion of our webinar. To our audience, if you have any questions you'd like to ask, please do so now. Just simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's get started. We have some great questions that have already come in. Konstanze, I'd like to start with you on this question. For this project, you investigated a prefixed community. What will happen when you monitor a growing community? Did you expect large shifts in the flow cytometry signal as cells move along the growth cycle?
Yes, happy to take the question. I guess this comes from the idea that maybe, as we saw with one of the strains, maybe there might be changes in the scatter signal, or depending on the strain used, maybe you might also see changes as the cells accumulate more DNA or RNA when they grow. So I think with the DAPI, we address some of these things, because DAPI, it's really independent of the cell activity, as far as I understand. With the scatter, I think it might be possible that you see that one community is not always in the same place as we had it here with the fixed setup. So in that case, maybe I would start with just monitoring one strain as it's growing along the cycle. But maybe Juan, you can add some comments as well.
Yes, that's the good thing that DAPI offers. DAPI is a dye that has high resolution. This allows to also monitor how the DNA quantity in each cell changes, and this allows us to monitor how this cell progresses in a growth cycle.
Thank you both. Now, while we're talking about DAPI, what do you think? How complex of a community will you be able to track with the DAPI staining? 3 strains, 30 strains, 300 strains?
Yeah, I think the 300, it's very ambitious. But maybe Juan, you can also say something about this. I guess you chose 3 for a reason, or your group.
We only chose 3 in these experiments because we are just validating a new procedure. But in our group, we had natural communities that have above 30 members. So it all comes down to the community you are measuring. There are some communities that will have members that are more similar to each other. This makes it more difficult to discern between one another. But we have had communities that we can identify until above 50 different members.
Yeah, that's super cool. Yes. And then you know that because you grow them separately, or how do you, or do you use other like molecular techniques to?
No, this was also done with DAPI staining, and they are not grown individually. These are communities that we collected from wastewater treatment plants. And we grow them in our lab in a synthetic wastewater medium, and we analyze ecological parameters of this community, also with DAPI staining.
And you can resolve 50 different, that's what you say.
More than 50 different sub-communities inside the same community.
Yeah, I think that's super cool because there are not many methods that can do that with environmental communities. No. Yeah.
Now, Juan, Konstanze, have you tried more complex communities?
For this procedure, not, because we only validated this procedure. We only want to standardize this procedure. Is the proof of concept that this DAPI staining can be done online and automated by the OC300. So in this case of this procedure, we have only used this small community that we presented.
Thank you. Our next question is 2 parts. How can you differentiate fluctuates in community composition noise from actual responses to a treatment? Can flow cytometry help with that?
So let me try to, sorry, can you read it one more time? How can you differentiate fluctuations,
Maybe the fluctuation, right.
In community composition noise from actual responses to a treatment. So I guess this is then going into once you have an online system running and you might, change the way you run, for example, your bioreactor process. So I guess the change would have to be significant enough. So I don't really know community composition noise, what that would be. So we see at least with this community that we tried here, that we have fairly stable signals. So any change outside this kind of more controlled experiment, I guess you would say it comes from a treatment, or maybe it could also come from ecological interactions within the community that change something about it. But I think the noise itself, I would not expect to be so big. I think that when you see changes, they come either from within the system or from a treatment. I don't know, Juan, what do you think?
Yeah, most of the times, a bacteria or a strain in a community will appear like in a Gaussian distribution. So it's easy to determine this from any background noise or any noise that you have in your sample. This normally will not distribute in a normal distribution. So it's easy to differentiate what is a bacterial strain present in your community from what is not.
Very good. Thank you both. Now we have a question asking, why do you use DAPI? Could this work with other dyes?
Mhm. Well, in general, you can use other dyes. I mean, the online protocol, it's very versatile. You can program it in other ways. We've, for example, worked with dyes like Cyber Green. Likewise, the Cytoflex, I mean, it has many channels, so you could use other dyes. I think DAPI brings a lot of benefits. Maybe, Juan, you can talk about that because your group really focused in on DAPI for this specific application.
Yeah, for this specific application, we choose DAPI over other stains because DAPI has a very high resolution. This allows for better separation between different sub-communities of the bacterial community. And for instance, in the case where you have more than 50 members in one community, if you use a stain that does not have this high resolution, you will not be able to resolve all the members of this community. So DAPI gives more resolution power in this case. That's why we chose it.
For community composition, yeah. So I think that's also something that goes back to that first question. When the cells grow, there are some stains that also stain RNA, and then cell activity will really mess up your signal in that way.
Yeah,
And they're really cool. Yeah, sorry, go ahead.
DAPI is exclusive to DNA. This is the difference to Cybergreen. Cybergreen is also widely used, but it also stains RNA, so this gives a poorer resolution.
And there are really cool stains out there. If your question is something else, for example, lipid accumulation could also be quantified with a different type of stain.
Yeah.
And you can do double staining also with the online method. So you could first stain for the community composition and then use a stain for something else and combine these methods.
Thank you. Now, Juan, we were talking about strains, 330, 300. Why did you choose these 3 strains to make up your bacterial community for the experiments?
Yes, these 3 bacterial strains are, I chose these 3 bacterial strains because these are bacterial strains that we used a lot in our working group, that we have a lot of experience with these strains. We call them mock community because it's an artificial community that we made. And we chose these 3 bacteria because they have different characteristics. 2 are one is gram negative, 2 are gram positive. They have different GC quantities in their DNA, and they also have different DNA quantities. So whenever this mock community we always use as our biological standard. Whenever we want to try a new procedure or a new device, we always come back to these 3 strains because we know how they behave and how they should look like when you measure them with flow cytometry.
Thank you. Now we have time for a few more questions. Juan, I'm going to stay with you for this next one. Can you use the OC300 unit only with the flow cytometer used for these experiments, or can you couple it to any flow cytometer?
I think Konstanze is better suited to answer this question.
Yes, because it's basically about the OC300, which our startup produces. So you can use it with other flow cytometers, not with any. I mean, it's always about coupling different machines, and the Cytoflex, it has a very nice interface to use with our system. And I also have to say that among the flow cytometers you can use with our system, the Cytoflex has the biggest, visual capacity, let's say, in terms of channels and combinations. But yeah, in principle, you could also use it with another one.
Very good. And I think we can wrap with this final question. It's come in several times, so people are wondering what's next. What are your next experiments that you have planned with the online flow cytometry setup? Do you already have insight into how well the system is working with samples that are more real life than in the mock community?
Yeah, I can just say from our side, from the whole automation perspective, I mean, we are working a lot on working with really dense communities, maybe more challenging communities like, I don't know, wastewater. You might think, that's challenging to look at in a flow cytometer without sample pretreatment. So that's something we work from the automation side. But maybe from the sample side, I think that would be with Juan.
Yeah, we intend to use our new procedure as a way of a sensor of bacterial communities that grow in a bioreactor. This new procedure gives real-time information, and we plan to use this in biotechnological procedures that use bacterial communities as main producers. So we are right now doing tests with these bioreactors and trying to expand the capabilities of this procedure.
Juan, Konstanze, thank you so much for your time today and for your important research. We do want to thank our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. And before we go, I do want to thank the audience for their excellent questions and for joining us today. Now, there were several questions we weren't able to answer, and those that are submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. I also want to draw attention to our audience for our survey in the corner. If you would like to speak to a flow cytometry expert at Beckman Coulter Services, my apologies, Life Sciences, please click yes or no in the survey on your left, and we'll have somebody reach out to you. Now, today's webcast can be viewed on demand, and Laboratory will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Thank you, friends, for joining us. We hope you all have a great day and hope to see you again soon. Bye-bye.